Greetings to everyone. My name is Kashish and I am pursuing my master's degree in bioinformatics at Amity University, Uttar Pradesh, India. This presentation is about my research fellowship at Pine Biotech on in-silico analysis of transcriptional responses to UV and IR radiations in skin cells. We all are aware about the harmful effects of sun radiations on our skin. So, to understand the DNA damage caused by these radiations, we use microarrays. Using microarrays, transcriptional reactions due to various conditions are studied. In our case, it's IR and UV radiation. These radiations affect our cell cycle, stress responses, body's cell repair mechanism, and many other functioning. In the dataset of our interest, microarray experiments were conducted on 10,000 genes from 15 healthy individuals with no history of carcinoma. These cell lines were exposed to radiations and data was recorded. This was a brief about the experiment that was conducted. Let's move to the pipeline we followed for our analysis. This slide gives us a gist about the followed workflow. Upcoming slides will discuss briefly about these steps, and finally we will move to the pictorial view of the research. An appropriate dataset was selected which sufficed our interest. The data was downloaded and filtered according to our requirement, and unwanted information was removed. The data, that is the expression table, was then grouped according to the tested samples into three major categories, namely IR, MOC, and UV. Once the data was grouped, various statistical analyses were performed on it, particularly PCA, which is principal component analysis, and manual calculation of p-adjusted, p-value, and fold chain calculation was done. Moving forward to pathway analysis of the genes, iLinks and PantherDB were used. In bioinformatics, pathway studies play a huge role in determining the key genes or proteins that hold importance in the trial or pathogenical conditions under study. They also help in better understanding of the mechanism of interest. iLinks is host to many pathway analysis tools which with many of their redirecting links attached with them. Panther databases provide us with its own benefits of graphical representation of functional pathways along with their evolutionary analysis. Now let's move to the pictorial representation. This slide shows the series matrix data that we downloaded along with the group data. This is the group data. After grouping, this is how it looked, with red representing IR, green representing mock data, and this blue color representing UV data. This picture shows the David result page of gene list, which was obtained by submitting the probe IDs of the dataset. Using R, the gene IDs were merged with the probe IDs and are presented in this picture. T-Bio platform is a cloud-based package of big data analysis application. It offers numerous pipelines to run our data and get the analysis done. It also gives the user an option to go through the demo pipelines for better understanding of the functionality of platform. Here PCA results are shown before grouping was done. It was done to get an idea about the distribution of the samples expression before grouping as well. After grouping, three PCA were run. They were mock versus IR and UV, mock versus UV, and mock versus IR. Both the PCs, in both the PCs, nothing much of significance was found. The samples were forming a scattered carpet of plot. Thus, coming to a conclusion was difficult. So we move to the pathway analysis. This picture shows us the iLinks homepage with various features ready to be explored. This is the signature submission page where we submit our signature, and this signature includes the gene IDs, the fold change value, and p value as optional category. After hitting submit signature, this output is recorded. This shows the signature analysis tools we can perform and get the information about pathways, networks, or general visualization of the genes. For this presentation, I have included the example of Enricher. Once the data is submitted on Enricher, this is the page that opens up in front of us with our gene results. Here also we have different pathways available for us to explore. I have opened KIG 2021 human bar graph. This shows the expression level was high in cell cycle followed by other pathways. We can also use the clustergram view to view the genes versus the pathway graph. It shows the genes here and their expression level on various pathways. Panther DB. This is the page where we submit the gene list for gene list analysis. These list is submitted by a comma separated file, and we select the options from here like Homo sapiens and select the analysis we want. If you want if you want to view the gene list, this is the result that is shown. And for graph, we select the graph option to have a graphical chart representation. This is a graphical chart representation of Panther DB pathway. This is the pathway between IR and mock, and there are many peaks in it, which we'll discuss further. This is the UV versus mock functional classification in bar chart, and these are the color codings of various pathways. uh, Results. Now in results, we have many top prop sets. From out of those prop sets, the majorly that were explored were stress response, cell cycle proliferation, cell structure, signal transduction, apoptosis, energy metabolism, and few other were there. The downregulated pathways showed more expression in contrast to the upregulated pathways. While going through the UV responsive genes, it was observed that around 59% of the genes were miscellaneous, as there was not much information that was explored about their role in radiational effects on the skin. Nonetheless, significant number of genes were selected, which has the relevance to the query pathways. IR and UV genes showed similar distribution in functional pathway. From these results, we interpreted that uh, if there is an due to the exposure to the radiation to the cell lines, there were abrupt activities of cell line of cell cycle and energy metabolism that caused skin lesions, and which may further lead to carcinoma development. Apoptotic cycles showed an increased expression, which may be inferred as uh, a way in which the cells, the components are aiding in prevention of further proliferation of the abnormal cells, or there is a breakdown of essential cellular components. The outcome proposed that protein translation is additionally hindered by halted transcription of tRNA synthetase genes. These were the references. This was the paper from which the dataset's main analysis was done, and followed by some other references. At last, but not the least, I would like to thank my mentors and Pine Biotech faculties for helping me in my fellowship. Have a great day ahead.